is Chris Carlson, and I'm a graduate student at UCSF in the Morgan Lab. The Morgan Lab traditionally studies cell division and chromosome segregation, but today I'm going to talk about a very different story I've been working on for the last year related to the coronavirus. So here you can see on the left is my cartoon depiction of the virus. The spike protein is shown in orange on the outside, but what I'm actually interested in is the inside of the virus. Specifically, I'm interested in how the virus packages its large RNA genome. This is a rather interesting question, as coronaviruses have some of the largest known RNA genomes among all RNA viruses. So how does it take that large genome and compact it down to fit inside the virus? And also, how does it protect its genome while it's in the air transmitting from one person to another? So we know a little bit about this. We know the N protein, or the nucleocapsid protein, is required for this compaction, but we really have no molecular understanding in how the N protein actually takes viral RNA and compresses it into a smaller state. So I went ahead and purified the N protein, and I purified viral RNA, and I mixed the two together and looked at them under the microscope. And you can see here on the left, it doesn't look like much. They look like kind of amorphous kind of blobs, right? But these blobs actually represent kind of jello-like compartments that are composed of N protein and viral RNA. And we believe this jello compartment may actually reflect the kind of matrix inside the virus. And this kind of made sense to us because the jello compartment may serve as a protective kind of, like I said, matrix to protect the RNA genome. We actually zoomed in a little bit more on these jello blobs as well to see if there's any underlying structure. And to our surprise, we observed these kind of discrete little particles, which you can see in the middle. And then after a little bit of data processing and biophysical characterization using other techniques, we kind of came to the conclusion that these particles likely contain 12 N proteins that have come together in a distinct manner and are surrounded by 600 bases of viral RNA. So this was quite exciting to us because this could represent kind of like a beads on a string model, where you have a bunch of these kind of particles or beads on the string of the genomic RNA. And these beads would allow for kind of wrapping of the RNA around them, which would allow for compaction of the genome. And very nicely, there's a complementary study that used another high-end microscope to look at the interior of mature full viruses, coronaviruses, and they actually observed very similar packaging units, which you can kind of see here in the middle. And so this led us to this model, which we have now on the left, where we believe the coronavirus genome is actually speckled with a bunch of these little packaging units. And right now I'm trying to get a higher resolution structure to understand how this occurs at the molecular level.